Okay, Professor Tabs is back. And remember, we are talking about antigen presentation. In particular, we're talking about B and T cell receptors. So uh, remember that there is a difference between the B cell receptors and the T cell receptors. So remember that for B cell receptors, it is the antibodies we're talking about. For B cells, these are the antibodies, and that they are receptive for a wide variety of macromolecules, whereas for the T cells, they can only see peptide fragments of protein antigens. So we're talking about amino acids here. It can only do so when these peptides are presented by specialized peptide display molecules on host cells, as we were just talking about the antigen presentation cells, the macrophages and the B cells and the dendritic cells. But B cells are very different. They use antibodies to recognize the antigen. So let's think about the antibody. And we've talked about this before in detail. So just as a quick review, we know that we have light chains and heavy chains, but it is the antigen binding site is the variable region, which is part of the light chain and part of the heavy chain. So moving on, let's talk a little bit more about the antigen receptors of the lymphocytes. Actually, let's talk a little bit more in detail about the antibodies. Remember that the antibodies may be membrane-bound antigen receptors of B cells or secreted proteins, and that they're also known as immunoglobulins. And we're going to talk about the isotypes of the immunoglobulins later. Remember that we have IgA, IgD, IgE, IgG, and IgM. And we definitely need to be very familiar with these to do well on the test. Anyway, continuing on, remember that for B cell membrane-bound antibodies, these recognize antigens to initiate the response, whereas for secreted antibodies, these neutralize or eliminate microbes and toxins in the effector phase of humoral immunity. So they're either connected or they're loose. When we say they're either membrane-bound or they're secreted, so that is the way to remember them. And again, remember the antibody structure and the binding site, and that is in the variable region. And the structure of the antibody, we have the light chains on the outside, and then in the middle you have that heavy chain that runs up on the inside of the Y, and there is a hinge. And that generally the antibody is composed of 4 peptide chains, including 2 identical heavy chains and 2 identical light chains, with each chain containing 1 variable region and 1 constant region. So the one part of the variable region is the Fab section, and the constant region is the FC region. So now we're going to talk a little bit more about the immunoglobulin isotypes. We're talking about the antibody heavy chains here, and there are 5 types. Each type of heavy chain may complex with any type of heavy chain. I'll say that again. Each type of light chain may complex with any type of heavy chain. And the isotype antibodies that contain different heavy chains, which is what makes them different. So what are the different antibody heavy chains? What are these isotypes? We have IgM, IgD, IgG, IgE, and IgA. Remember that each isotype has a different physical and biological properties and effector functions. So let's discuss what they are in general. First, how about IgA? Can you remember what the function of IgA is? The isotype of this antibody is IgA. And we don't have to talk about the serum concentration at this point or the half-life or where it's really secreted from, but we should realize that the function of IgA here is mucosal immunity. So we can think about the GI, urogenital, respiratory, all the mucosal areas. Let's think about the lungs, for example. Think about immunodeficiencies. Breast milk. IgA is secreted in breast milk. So think about IgA and mucosal immunity. How about IgD? We don't know a lot about IgD, except that we do know that it is a naive B cell antigen receptor. And How about IgE? So IgE, when we think about IgE, we should think about the function is for mast cell activation, and this is for type 1 hypersensitivity and defense against helminthic parasites. So those that should be quite easy to remember. So IgE, we have type 1 hypersensitivity, mast cell activation, defense against helminthic parasites. So how about for IgG? And this is the antibody which is present in chronic infections, and IgM is present in the acute response to infectious microorganisms. Anyway, talking about IgG, a very busy antibody. Has a very high serum level, and when you think about IgG, think about the opsonization of complement activation, antibody dependence, and cell-mediated cytotoxicity, neonatal immunity, and feedback inhibition of B cells. So IgG, neonatal immunity. Why? Because it crosses the placenta. And let's think about IgM. So IgM is present for an acute response to infectious microorganisms, and it's similar to D in that D is for naive B cell antigen receptor, and also this is also a naive B cell antigen receptor, and is used in complement activation. So let's just quickly run through this again. So when you think of IgA, what do you think of? Mucosal immunity. IgD, naive B cell antigen receptor. IgE, mast cell activation, immediate hypersensitivity and defense against helminthic parasites. IgG, opsonization, complement activation, antibody-dependent cell-mediated cytotoxicity. Neonatal immunity, feedback inhibition of B cells. IgM, naive B cell antigen receptor complement activation. So here's an MCQ for you. CD4 T cells recognize peptide in the content of MHC class 1 or MHC class 2. The answer is B, MHC class